Hello everyone, and welcome back to the AI Research Roundup. I'm Alex, and today is May 6th, 2025. Alright, we're diving into this ICLR conference paper, GeSubNet, Gene Interaction Inference for Disease Subtype Network Generation, which came out just over 3 months ago. So this paper addresses a key challenge, understanding how genes interact differently in various disease subtypes. It proposes a framework using tools like VQ-VAEs and graph neural networks to integrate patient data with existing gene knowledge. Okay, so this first figure really helps visualize that core challenge. On the left, you see a general breast cancer gene network, like one you'd get from a database. But the issue, shown on the right, is that this broad view doesn't tell us which interactions are truly active or important in specific subtypes. Say, luminal A versus basal-like. It highlights this mismatch, making it hard to understand subtype differences just from general knowledge. Alright, so building on that challenge, this next figure lays out their proposed solution, the GeSubNet framework. It's got 3 main parts. First, patient M uses a VQ-VAE on patient gene expression data to learn distinct subtype representations. Then, Graph M takes prior gene networks from databases and learns their structure using a GNN. Finally, Inference cleverly integrates these 2 representations to actually generate those subtype-specific gene networks we need. Okay, moving to the results. This table, Table 2, shows how GeSubNet stacks up against several baseline methods. They tested it on 4 different cancer datasets, using metrics like graph edit distance, delta-con similarity, and coefficient of degree variation. Well, the key takeaway here is that GeSubNet, highlighted in bold, consistently performs the best across all datasets and most metrics, suggesting it creates more structurally distinct and meaningful subtype networks compared to previous approaches. Okay, so after seeing the structural performance, Figure 3 delves into the biological relevance. These Venn diagrams compare the gene ontology terms identified by GeSubNet and the baselines across the 4 cancers. Notice how GeSubNet, shown in yellow, consistently identifies unique biological functions like immune responses or tumor necrosis factor signaling, listed below. This really reinforces that the generated networks are not just structurally different, but also capture more biologically meaningful insights compared to the others. Building on those unique biological functions we saw in the Venn diagrams, this next table, Table 3, actually counts them up. It shows the number of enriched biological functions, EBF, identified by each method with high statistical significance. Well, you can see GeSubNet consistently finds the highest number across all cancers. For instance, 8 in BRCA. This really backs up the idea that its generated networks capture significantly more functional diversity and biological relevance. Okay, so beyond just counting functions, Figure 4 tests the impact of genes identified by GeSubNet. Part A shows a UMAP visualization, knocking out high-ranking genes identified by GeSubNet, shown on the right, causes a significant shift in patient distribution, unlike low-ranking genes on the left. Then, Part B quantifies this shift rate. GeSubNet achieves a much higher rate, 0.83, for its high-ranking genes compared to baselines, validating its ability to pinpoint truly impactful genes. Alright, so after establishing the impact of the identified genes, Figure 5 gives us a direct visual comparison of the networks themselves. It shows the gene networks generated for 2 different BRCA subtypes, by GeSubNet on the left and 2 baselines on the right. Well, you can clearly see larger structural differences between the 2 subtype networks from GeSubNet, whereas the baseline networks look much more similar. This visually confirms the quantitative results we saw earlier. So, to wrap things up, we've seen quantitatively and visually how GeSubNet generates these distinct, subtype-specific gene networks. It really excels at integrating patient expression data with prior knowledge, producing networks that are not only structurally unique but also biologically relevant, pinpointing key genes impacting patient subtypes. It's a powerful approach. Well, that's all for this AI Research Roundup. Thanks for tuning in, and see you next time.